Hello, this is our lesson for AP Biology from Unit 2 on cell transport across the membrane. So the first thing we talk about are the actual things that the cell needs in order to survive, in order to maintain homeostasis. So it needs all the macromolecules that are the building blocks for other parts of the cell. So lipids, carbohydrates, proteins, nucleotides, and they get that from food if they're a heterotroph. If they're an autotroph, they can synthesize the macromolecule from carbon dioxide and water, along with the other dissolved nutrients, phosphates, and nitrates from the environment. Water is going to be used for synthesis to maintain their cell structure and dissolve materials. Oxygen is going to be used for respiration. And then electrolytes, free ions like sodium, potassium, iron, magnesium, calcium, are going to be involved in making the complex molecules function and in cellular communication. So these are all the things that the cell needs to take in order to maintain its homeostasis. So what allows it to take in certain things? The main thing that's going to determine whether or not something can go across a membrane, because if you remember, the membrane is made up of mostly phospholipid, is the molecule's lipid solubility. So ions like potassium and chloride, they need to move across at a rate higher than the lipid solubility warrants, so they're going to do so via special channels made up of proteins. Most biochemical molecules are too large or hydrophilic to pass through the membrane by simple diffusion, and therefore they require specific membrane proteins for transport. So I have a diagram that demonstrates which type of molecules can go through. So interestingly, urea and ethanol, they are actually polar molecules, but they have the ability to transport through the membrane, as does water. Small non-polar molecules, CO2, nitrogen, and oxygen, can easily pass through, but larger polar molecules, amino acids, glucose, ATP, they cannot pass through the membrane. Same thing with the electrolytes, phosphate, carbonate, chloride, calcium, magnesium, and potassium. They all cannot pass through the lipid bilayer. So things that can pass directly through the lipid bilayer do so via passive transport, or also just called simple passive transport, or just simple diffusion. If they have to pass through a protein channel, we call that facilitated passive transport. So again, usually only small non-polar molecules can travel through the lipid bilayer. Some ions in water will diffuse through channels. Again, water can travel through the membrane, however, it doesn't do so very easily. So membrane transport proteins are integral proteins that span the membrane and are utilized for transporting specific molecules or ions. So there's two categories of membrane proteins. There's uniports, which transport a single material, and then co-transporters, which will transport two materials. The co-transporters can be a symport, meaning both things are transporting at the same time, or an antiport, meaning the solutes are moving in opposite directions. So one thing will enter the cell while another thing is moving out of the cell. I show a graph here, which is typical of something that has to do with solute concentration, and that is there will always be a saturation point. So the rate of diffusion of materials through channels will reach a saturation point once you actually get to a particular point. So if you think about it, there is a certain number of channels, and only so much can actually travel through the channel at a time. So therefore, once you have increased the amount of materials there, you can't get through it anymore. So the rate at which the increase stops, and it basically gets to a plateau. And that only happens in facilitated diffusion. There are going to be limits to how fast simple diffusion can happen as well, but that's going to be more based on the size of the cell. Note that passive transport materials are moving down their concentration gradient, which means they're going from areas of high concentration to low concentration. Active transport generally means that materials are going to be moving against their concentration gradients, meaning going from low to high, as this diagram illustrates. And as this says, the active transport is also going to be expending energy, so it's going to be using ATP. So let's talk about the actual mechanisms, the actual way in which things move. So diffusion is natural movement. The one thing that's always kind of confusing for students is what is actually causing the movement of materials. And even at a solid state in a solid material, there is going to be some movement at the molecular level. So natural molecular movements, and think about the reason why that molecules will move naturally. It's because the thing that's holding the materials together are electrons, which are in constant motion. So the constant motion of the electrons are going to cause the molecules to wobble, to vibrate. And the only reason that they would not vibrate is if they're at absolute zero temperature, 0 Kelvin. So everything's going to be moving. So naturally, that movement is going to cause collisions. And as collisions occur, then they will dissipate until fewer and fewer collisions occur. But still, collisions can continue to occur. And collisions are more likely to occur when they're in higher concentrations. So as it dissipates, there are fewer collisions, and therefore the concentration ultimately reaches a point of what we would call dynamic equilibrium. So the next question we have is, what factors affect the rate of diffusion? So at this point, you should pause the video and think about those on your own. See if you can jot down some possible ideas. So the rate of diffusion is going to increase with temperature. So the hotter it gets, the faster the rate of diffusion. A decrease in density. So that would mean, like, if the fluid bilayer, the lipid bilayer, is less dense, a steepness of the concentration gradient, meaning if there's a higher concentration of solute on one side than the other, it's more likely to move through more quickly than if the difference between the two concentration gradients is not very high, or the two concentrations isn't high. Therefore, it's a lower concentration gradient. The size of the molecule and the electric charge of the diffusing molecule will also have an influence on the rate of diffusion. So certain things will move more readily across the membrane than others. Surface area to volume ratio is the key in how fast something can move, and also ultimately determines how big a cell can get. So very, very tiny cells will have a higher surface area to volume ratio than larger cells. And as a result, more materials can diffuse through those cells. Next, we'll talk about osmosis. So osmosis is a specific type of simple diffusion. In many cases, it's a type of facilitated diffusion if water's traveling through a channel called aquaporin. And osmosis is the diffusion of free water. And what free water is water that's not interacting with other materials. So in most cases, water is not going to be pure or distilled. There's going to be some solutes in there, and the solutes are going to be dissolved in the water as a result of the water having a polar charge and interacting with other things that are polar. So solutions with equal osmotic potentials are isotonic. A hypotonic solution has a lower osmotic potential than the other solution, and a hypertonic solution has a greater osmotic potential. We'll explain what those mean. But if you look, the osmotic potential is going to be relative to the concentration of the solutes. So this is a picture of a cell in a hypertonic solution. So relative to the inside of the cell, there is a higher solute concentration. What's going to happen is the water is going to travel outside of the cell. Inside the cell, or as a result of that, the membrane would shrivel up. This is actually referred to as plasmolysis. In a hypotonic solution, there's going to be a lower concentration of solute outside. Hypo means low. So what you'll see is water will travel into the cell, and in many cases, it can cause the cell to explode. That's called cytolysis. Finally, in an isotonic solution, you'll have equal amounts of solute on both sides of the membrane. There would be no change in the membrane. So here is a diagram just referring to the percentages. So if you have 5% solute on the outside, 10% solute in the cytoplasm of the cell, what that assumes is that you have 95% free water outside, 90%. So this is the one thing that's kind of confusing about osmosis, because usually thinking high to low, but in this case, water is going to flow from hypertonic to hypotonic, from high to low solute, because again, what we're talking about is the movement of free water. So therefore, it's 95% free water going to 90%. So when you see the solute concentrations, generally what you want to focus on is, well, then what would then be the concentration of free water? So if the concentration outside the cell is 20% and the inside is 10%, the free water concentration inside the cell is 90%, whereas outside is 80%. So it will go from high to low, and therefore this cell would lose water. Okay, so the black numbers are the solute concentration, the red numbers are the free water concentrations. So before I talked about osmotic potential, so what does that mean? So osmotic potential, also just referred to as solute potential, it's a measurement of the likelihood of the water to travel from its solution into a new location. And so it's always a negative number. And basically osmotic potential or solute potential is it's a type of vacuum pressure. It's measured in bars. And what's happening is, as it says, as the solute concentration goes up, the higher the osmotic potential. And when I say higher, I mean more negative, meaning that solute or that solution will more likely take up water. Osmotic potential is a component of water potential. Water potential takes into account pressure potential, which is the exertion of pressure on the solution, but usually we consider that negligible in animal cells because the pressure on either side of a cell, outside and inside, are usually equal. So therefore, solute is going to be the thing that makes up that. We generally discuss water potential in transpiration, the air and the air pressure and the inside of the pressure inside of a tree or inside of a leaf are going to be different. So therefore, the movement will be affected more by pressure potential. But osmotic potential is something that you would need to know how to calculate. Now, you would be given the formula, and the formula is here. It's psi sub s for the potential, and then the S stands for the solute, equals negative ICRT. Again, it's negative because the type of pressure is a vacuum pressure. So the lowest potential, or I guess you could say the highest osmotic potential, is 0. And that would be pure water, distilled water. And that's because materials aren't going to move into a solution with no solutes. It can only move out of the solution with solutes. Then the I is going to be a value related to the solute. It would be 0 if it's nonpolar. So obviously nonpolar isn't going to affect the osmotic potential. 1 if it's an organic polar molecule, and 2 if it's an ion. So if it's a salt, you would put a 2 there. C is then going to be the isotonic concentration. You might also see that referred to as the osmolar concentration. and it's a concentration in molars. Molars is moles per liter. So if you don't know what moles are, make sure you look that up. It's a number that has to do with the atomic mass of all of the elements that are in the molecule. And if you add all that up, add up all the atomic masses and then convert that to grams, that's going to refer to as 1 mole of that substance. And it's going to be relative to a particular number. So if I was creating, say, a mole of salt, where salt is going to have is NaCl. So Na has a atomic mass of 23, and Cl has the atomic mass of 35. So 23 and 35 added together is going to give you 67, or 57, sorry. And that would then be the number of grams that you would then dissolve into a liter to get a single molar of salt. So R is a constant. It's 0.0831 liter bars per mole K. So basically you use that to get down to the unit that you want. And then the temperature would be the temperature in Kelvin of the system. So Kelvin is degrees Celsius plus 273.15. So after you calculate something, you ultimately will you ultimately eliminate all the units other than bar, and again, bar is a unit of pressure. So this is an example problem for this, and we will actually do this one in class. So I'm going to leave this for to stay here so that we can go over this in class. But this is a sample of a osmotic potential problem that would have come from an AP exam. So osmotic movement of water in cells lacking walls depends only on the osmotic potential between the cells. So one of the things that you see here, this is a protistin. They have walls, but their walls are pretty flexible. So they have to actually pump out the excess water that they constantly take in when they live in a fresh water environment. So they use what's called a contractile vacuole. So what you see here is a contractile vacuole filling up, and then it has little channels that as it then squeezes shut, it closes, it shoots out the water out of the paramecium. Active transport is the next type we're going to talk about. So active transport is where a carrier or a pump is going to be used. So a lot of times the pumps are going to be called ATPases, or they're going to have an ATPase subunit as part of it. And what this is right here, the phosphate bonding site, it's going to be an ATPase subunit. And ultimately what happens here is ATP is going to be is going to interact with this protein, and a phosphate is going to be given off. So if we remember about phosphate functional groups, they're very negatively charged, and when it attaches to the protein, it causes the protein's shape to change. So that's what's being represented here. The shape or conformation of the protein changes, and that allows the material to move across. So it physically causes the protein to change, and therefore move the material that attaches to it. So usually you have an attachment site for it, a solute, and then it's going to be able to move it across it after it attaches. So once the phosphate detaches, it goes back to its original state. So you would, start back over it at step 1. So it kind of works as a pump. It opens up, it closes, opens up, closes, opens up, closes, and it continues on. And every time it opens up and closes, it has to gain a phosphate and then lose a phosphate. So it uses an ATP. So active transport is one of the main things that you use ATP for. So much of your calories that you are burning in a day, somewhere in the neighborhood of 60%, are going to be used just for active transport. And we'll talk about why you need to do active transport later in future in future lessons. So there's three types of active transport. There's the primary active transport, what we just demonstrated. Then there's secondary active transport. So secondary active transport is interesting. It uses a primary transporter to create a concentration, usually of ions, and then you have a symport. And the symport will only work to move a target solute through the channel if also the ion is moving through the channel at the same time. So a lot of times it's considered voltage gated, because what happens is when these ions build up a high enough concentration, it changes the voltage outside the cell. So that voltage causes the whole thing to kind of change in shape, and therefore everything can move through. So once there's a gradient of this ion, then this solute can move through. Then there's coupled active transport, which is where you're moving two things simultaneously. And the famous example of coupled active transport is called the sodium-potassium pump. So this is a sodium-potassium pump. So what you see here is in step 1, sodium bonds to the attachment site there. Then ATP is going to be broken up into ADP. So ATP is going to attach a phosphate group. That's step 2. As a result, the sodium-potassium pump changes its shape. So you can see the bonding site now went to here. So it basically flipped. And then potassium here is going to attach. Only 2 potassium attach per every 3 sodium. Then what happens is the phosphate group is detached. So that's down here. So the sodium is going to be released. The potassium will attach. Phosphate group detaches. It goes back to its original conformation, and then the sodium now can be released into the cytoplasm of the cell. What ultimately this does is it creates a gradient that can be harnessed in various ways. One of the ways in which it's used is here in the intestines. So this is a sodium-potassium pump right here. And what it's doing is it's taking, here's the blood, and it's taking glucose here. And what it actually wants to do, it wants to start with the glucose here in your intestines, and it wants to get it out to the blood. So you see, ultimately what this is showing happening is glucose going from inside your intestines getting into your blood, because the blood's going to then transport that glucose into your cells. So how is it doing that? Well, glucose is going to travel by facilitated diffusion, but it's also going to travel through with sodium. So this is kind of like a secondary active transport. This is a glucose transporter, but it requires that sodium travels through as well. So sodium, the glucose is going to have to go from high concentration to low concentration. Sodium is going to go out or through here only when a low concentration of sodium is in here. So how do we maintain a low concentration of sodium? We pump it out. So if we constantly have a high concentration here, we need to pump the sodium out. So that's what we use the sodium-potassium pump to do. And then this glucose is then transport or go through this transporter by facilitated diffusion. So we'll talk about that in more depth in a later lesson, but this is just an example of how sodium-potassium pumps are used. Again, it uses it to kind of create a gradient so that naturally this stuff can actually move through. All right. Finally, there's a type of active transport called bulk transport. And there's 2 basic ways in which this happens. It's through endocytosis and exocytosis. The one thing to note about endocytosis and exocytosis is it makes it look like the membrane is doing everything independently. And in fact, what's happening is that there's going to be cytoskeletal elements. So if I show you what's going on here. So usually there's going to be some sort of cytoskeletal element that's going to be grabbing onto these membranes and actually transporting down using ATP these vacuoles. So a vacuole can form right here. So again, this would be grabbed onto by cytoskeletal elements, and everything is going to be orchestrated by various proteins and things like that to actually cause this to happen. So this enveloping or enfolding that you see here, it doesn't just happen naturally. It's going to happen in an orchestrated fashion controlled by cytoskeletal elements. In the same way that cytoskeletal elements will cause your muscles to move, these move around the vesicles. Exocytosis is the reverse of that. It's going to be when these materials are going to be attached, and again, they're going to be pulled to the membrane by little proteins usually that are going to travel along the cytoskeletal elements to allow the materials to attach and release. So that concludes our lesson on the mechanisms of movement in and out of the cell.